Hello, biology students, and welcome to this video where we're going to be discussing mitosis. So as we discussed in class previously, mitosis is the process by which cells divide. And in particular, we're going to be focusing on how the cell's nucleus divides its DNA. So if we kind of move on to the next slide here. The first thing I want to do is give you a little bit of background kind of information and vocabulary. So when we're talking about the nucleus dividing, we're often talking about the organism's DNA dividing. So therefore, it will be important for us to first establish what DNA looks like in a eukaryotic cell, that's a cell with a nucleus, versus a prokaryotic cell, a more simple bacterial cell that does not have a nucleus. So one of the first big differences between eukaryotic and prokaryotic cells is because eukaryotic cells are more complex, they just have more DNA than prokaryotes do. They have way more genes. So the example I gave you on the slide here is that human cells have about 25,000 genes in a single human cell, whereas a bacteria only has about 3,000. They have less DNA, it's simpler, they have fewer genes. And then the other big difference is what that DNA actually looks like. So if we're looking at a eukaryotic cell, that larger amount of DNA is kind of organized into these linear structures that we call chromosomes. So chromosomes are like a single piece of DNA. So if you were looking in the nucleus of a eukaryotic cell, you would see DNA organized into what looks like little lines. And each of these lines would be a single chromosome. We can contrast that with the fact that if we were looking at DNA in a prokaryote, because they have less DNA, they don't need multiple chromosomes. And instead, their DNA is organized into just one single circular chromosome. So they have just one chromosome, and then it's organized into kind of like a circle shape like this. So eukaryotes have many linear chromosomes, and again, chromosomes are DNA, whereas prokaryotes have only one circular chromosome. So if we kind of like think a little bit more deeply about what chromosomes look like in a eukaryotic cell, really a chromosome is just a way to organize a cell's DNA. So you can see in this picture over here, down in the bottom right, each of those blue squiggly lines is one of the chromosomes in this eukaryotic cell. And the way chromosomes are organized, I want you to imagine you had a big string of DNA. And in the cell, when the chromosomes are kind of made, there are these proteins called histones, which I'm drawing in these blue circles here. And then I have my really kind of big, long piece of DNA. So what the cell does is take this long piece of DNA and basically kind of wraps it around these histone proteins in this kind of organized fashion. And by kind of coiling up the DNA, you get that linear chromosome that we had drawn as a single straight line on the previous slide. So a chromosome is a really, really long DNA molecule wrapped around proteins, histones. When DNA was in that long, stringy, kind of unorganized form, we called that long, stringy, unwound form chromatin. And then when it's wound up and organized around the histones, it's called a chromosome. In the really wound up, organized form is really not a good form for the cell to access and use the DNA. So most of the time when the cell's just doing its job, the DNA is in its unorganized chromatin form, so proteins can read it more easily and use those genes to make proteins for the cell. However, when the cell is dividing, you want the DNA to be organized in a way that it will be easy to split it up between the two cells that you're creating. So just before cell division, that's when chromatin coils up and compacts into that chromosome form. Because chromosomes are the compact version, you can really only see the compact DNA in chromosome form when cells are actually actively dividing, when they're actually in mitosis. So I have a really, I think, funny cartoon here that has a little biology humor for you. So on the left, this big black squiggle is a bunch of DNA in its chromatin form. It's unorganized. It's not wrapped around histones. Where on the right, we have DNA in its organized chromosome form, that orange X. And he's saying, dude, like mitosis starts in 5 minutes. I can't believe you haven't condensed yet, right? So when the cell goes to divide, that's when the DNA is going to wrap around chromosomes to become more organized and more readily ready to divide during cell division. So a little bit more chromosome vocabulary for you. So here we have like an actual electron, like microscope picture of a chromosome, which is pretty neat. So we learned already that during S phase of the cell cycle, chromosomes duplicate, right? Like we make a copy of our DNA because we know we need 2 copies of DNA for the 2 sister cells or daughter cells that we're creating. What we didn't talk about in class yet was the idea that when DNA duplicates during S phase, the spot that I'm circling over here in blue, this left-hand side of this X, this was the original DNA that was in the cell before S phase. And then this half on the right, that was the copy that got made. And one thing the cell does is it takes the 2 identical copies, like the original copy plus the copy it made during S phase, and it keeps those copies kind of attached together in this X shape. So you end up getting one piece of DNA that was like there before S phase, and then the other side is the copy. So therefore, the left and right-hand side of this chromosome that we're drawing as an X are 2 identical copies of DNA, one for each of the daughter cells. So this point where those 2 copies are joined, we call that the centromere. And then each of the 2 halves, the 2 identical pieces of DNA, are called sister chromatids. So if you hear those vocabulary words, that's what it's talking about. So let's see what we got next here. So like I said, chromatids, that spot where they're joined together is called the centromere. And then that X shape, sometimes we call it a replicated chromosome because it is a chromosome that has replicated or copied itself. So you have a lot of vocabulary so far that sounds similar. You had chromatin, chromosome, chromatid, and centromere. All those words sound really similar, but they mean different things. So kind of getting those vocabulary words straight in your head is going to benefit you. All right, so if you think about like mitosis or cell division on the whole, really what's happening is during interphase, our DNA is going to duplicate. So we're going to go from a kind of an unreplicated chromosome up on top. During S phase, that DNA is going to duplicate. So now we have 2 identical copies. And then during mitosis, when the nucleus splits, each copy goes to one of the daughter cells. So you end up with 2 cells that both have identical sets of DNA. Once like those chromosomes have separated, the 2 sister chromatids have gone to their individual daughter cells, we now call that line a non-replicated chromosome. So a replicated chromosome is when it's in its X shape and it has the 2 identical halves. A non-replicated chromosome is when it's in its kind of like I shape and it only has 1 copy of the DNA. And as a result, we get 2 daughter cells that each have an identical set of DNA. So now let's go ahead and talk about the individual steps of mitosis. So we defined in a previous class that mitosis is the process by which the nucleus divides into 2 identical nuclei. And we have a goal of our 2 daughter cells being identical to each other, so therefore having the exact same DNA, identical copies of DNA. So when we're talking about humans, for example, we're talking about starting with a cell that has 23 pairs or 46 individual chromosomes, and then therefore we have 2 daughter cells after mitosis that would have 2 sets of 46 chromosomes. Now, different organisms have different numbers of chromosomes. So if dog cells were going through mitosis, you would start with a cell with 78 chromosomes and end with 2 daughter cells with 78 chromosomes. The important thing is that the daughter cells are identical to each other and to the original parental cell. So remember, I want to remind you that mitosis exists as part of the larger like cell cycle process. So remember, we had talked about that the cell cycle has interphase that has G1, S, and G2, and then we have the mitotic phase that includes mitosis, which is what we're talking about here, and also cytokinesis, the actual physical division of the cell itself. So when we're talking about the mitotic phase, that's really what we're discussing here, mitosis and cytokinesis. It turns out mitosis itself can be split up into 4 like even smaller phases. So mitosis itself can be split into prophase, metaphase, anaphase, and telophase. So we're going to walk through each of those steps individually, and then we'll discuss cytokinesis. So remember first, before any of that, we get interphase. So interphase had G1, where the cell grows, organelles duplicate, S phase, where the DNA duplicates, and G2 is where the cell grew even more. The goal of interphase, all these things happen so that the cell is ready to divide. We have enough cytoplasm, we have enough organelles, we have enough DNA for 2 daughter cells. So the first step of mitosis is going to be prophase, and that's going to immediately follow G2. And the first thing that's going to happen is that our replicated DNA is going to condense, it's going to organize itself from its chromatin form to its chromosome form. So we're going to kind of wind up our DNA so it's organized into that chromosome form and it's ready to be separated. In order to separate the nucleus, we have to get the nuclear envelope out of the way, which is the membrane of the nucleus. So that nuclear envelope also will break down and disappear. And we will also get the organelle that it's actually going to do the separation of the DNA is called centrosomes or centrioles. So that's this organelle in yellow here. And those organelles are going to migrate to the 2 opposite sides of the cell. And that organelle, the centriole, is going to build these little kind of yellow strings called centrioles. And those, they're called spindle fibers, my bad. And the job of the spindle fibers is they're going to kind of act as like lassos, and they're going to physically attach to the chromosomes and pull them apart when the nucleus goes to divide. So just kind of a vocabulary note here, the whole structure is called the centrosome, where the actual organelle that actually builds the spindle fibers is called the centriole. Okay, so here's a picture of an actual cell in purple here going through prophase. You can see the pinkish stained organelle are the centrioles, and then the dark purple squiggles are those condensed chromosomes, so where the DNA has condensed from chromatin to chromosome form. All right, another cell kind of showing the same thing. The dark black is the DNA, and that kind of lighter brown color is the centriole starting to form. Okay, so next we're going to get metaphase. So in metaphase, at this point, the centrioles are at opposite sides of the cell, and the spindle fibers that they've built have attached to my replicated chromosomes and align the chromosomes so they're all lined up in the middle of the cell, nice and organized in a single file line. So the centrioles are at opposite holes, and the spindle is completely formed. And then down the middle of the cell, what we call the middle of the cell, the metaphase plate, the chromosomes are all lined up in a line. And what happens is the spindle fibers are going to attach at that joining point at the centromere, the center of each replicated chromosome. And basically in the next step, they're going to get ready to pull them apart. I think that metaphase starts with M, and in this phase, all the chromosomes line up down the middle, which also starts with M. That helps you remember what happens in this phase. So we have some actual pictures of cells undergoing metaphase here. So you can see here is my cell. All these pink lines are the spindle fibers, and you can see my DNA has lined up in a nice line down at the center of the cell. All right, so here's another picture of a cell in metaphase. Again, that dark stain is the DNA, the chromosome lined up in a straight line down the middle of the cell. All right, so our next phase is going to be anaphase. And I think anaphase starts with A, and the word apart starts with A. So in this phase, the replicated chromosomes are going to be split apart. What's going to happen here is I want you to remember that each individual replicated chromosome, each X, that the left-hand side of that X and the right-hand side of that X were identical pieces of DNA. So what's happening in anaphase is we are pulling apart that X. We are pulling one half, the spindle fiber is pulling one half to one side of the cell and the other half to the other side of the cell. And because those two halves are identical, we're starting to get one set of DNA that's identical to a second set of DNA. So we're kind of grouping this DNA into two piles that will end up being like two identical groups that will go to two new daughter cells. So the spindle fibers pull apart the sister chromatids, and those sister chromatids we now call non-replicated chromosomes, like vocabulary-wise. And this works by motor proteins, basically pull on the spindle fibers. Imagine them like pulling in that fiber until the chromosomes are totally separated into two identical groups. So here are some pictures of actual cells going through anaphase. And you can see we're starting to get DNA kind of separated into two distinct piles here, our two distinct groups ready for our two new cells. Another picture kind of showing you the same thing. All right, so our last phase of mitosis is going to be telophase. And I think of that telophase starts with a T and two starts with a T. So we really are starting to form two nuclei during telophase. And telophase is basically the opposite of prophase. Everything that happened in prophase, the opposite is going to happen in telophase. So for example, we are going to get two nuclei reappearing around like each group of DNA. So we're going to get two nucleuses, two nucleoli, two nuclei, and two nuclear envelopes forming around my two groups of DNA. The chromosomes are done separating, so they don't have to be in their organized chromosome form anymore. So they decondense, they unwind back into their spaghetti, like unorganized chromatin form. And again, this is important because it allows like proteins to actually access that DNA and use it for things. And then the spindle fibers disappear because we no longer need them to separate the DNA for us. And at this point, mitosis or nuclear division is now complete. So again, some pictures of cells undergoing telophase. You can see two distinct nuclei forming, and that's how we know our cells in telophase. In another picture here. So then finally, mitosis is followed by cytokinesis. And cytokinesis is when the cytoplasm splits. An animal cell, this basically happens by the cell membrane pinching. And that pinching location is called a cleavage furrow, and this forms new cells. So you can see in this picture here, kind of that center where it pinches off, that's called the cleavage furrow. So we have a cartoon on the left showing you that. We have an actual microscope picture on the right, which is pretty neat. So here, that pinching location is called the cleavage furrow. And we get one nucleus in each new cell. And however, because plant cells have a cell wall, it's not quite as simple. You can't pinch that kind of rigid cell wall in a plant cell. So plant cytokinesis looks a little bit different. So first of all, when plants didn't actually ever have centrioles to build the spindle fibers, but the spindle fibers still existed. But the other big difference I want to talk about here in cytokinesis is that because the cell can't pinch off, and so we basically build new cell wall between the two new cells that are forming, and that new cell that we build is called the cell plate. So you can see down in this cartoon picture down at the bottom, instead of pinching the cell off, we just built a cell plate, built new cell wall in order to ultimately create two new cells during plant cell cytokinesis. All right, so to kind of overview the whole process, we started in prophase where we had the DNA condensed, we got rid of our nuclear envelope. In metaphase, the chromosomes lined up down the middle of the cell with the spindle fibers attached. Those sister chromatids were pulled apart in anaphase, and we created two new nuclei in telophase, and then ultimately divided the cell. So again, I have a couple pictures, actual photos and cartoons here showing you the same thing. So we have interphase, followed by prophase, where we condensed our DNA right into our chromosome form. And then the chromosomes lined up down the middle of the cell in metaphase, were pulled apart in anaphase, and finally created two new cells during telophase and cytokinesis. So that's going to be it for this video, and I will see you in the next one.